So, you've made it to segment 3, function 3. Hopefully this is not 10 o'clock at night on Sunday, January 7th. Hopefully you took advantage of doing this much earlier. Either way, welcome back to segment 3. So let's talk about the third job. Cells need to make more cells. Cells don't live forever. Cells don't last forever. I already mentioned certain cells only last a few days. Other cells might last years. But in many, many cases, cells have to be replaced on a regular basis. The only ones I can think of off the top of my head that are not replaced very often are nerve cells. That's why nerve damage and brain damage is very, very difficult to repair, because there's no new cells being produced. Most every other part of our body, cells are constantly making new cells. So let's go into this. So making more cells, well, why? Because we have to replace things when they wear out. We have to repair things when they get damaged. And for most of our life, we're going to grow. Even if we're not talking about your whole body, you still grow hair, you still grow nails, etc., etc. So first thing you got to do if you're going to reproduce is you have to copy your DNA. You're about to make another cell. Well, every cell has to get every cell has to get a copy of that original 46 chromosomes that were put together the night mom and dad conceived you. And every one of your several trillion cells has those same 46 chromosomes in them. So every time you make a new cell, you have to copy that full set of 46 chromosomes so the cell has the instructions for being you, particularly. You also will have to make extra organelles, because again, you're making a new cell. You can't start with no organelles. Organelles carry out all the functions. And then somehow you have to figure out how to divide up that DNA and organelles between the two new, what we call daughter cells, the daughter cells are the new cells, that is accurate. Because you want to make sure that each new cell gets one copy of every one of those 46 chromosomes. You don't want one cell getting extra copies. You don't want one cell getting shorted copies. They won't live. You want to make sure each cell gets enough of each organelle to carry out all the different functions it needs to do. So this has to be a fairly organized process. It can't just be haphazard. It has to be fairly organized in order for this to work properly. And the big part of this is what we call dividing up the DNA. So here we've got in this picture here a cell that is in the middle of dividing. This is something called metaphase. You've probably heard of mitosis before. We'll get into this second semester as well, so I'm not going to go into a lot of details on this today. But the biggest thing we have to divide is this DNA. Here we see 2 blue chromosomes, 2 red chromosomes. Maybe the 2 blue chromosomes are the ones you originally got from your dad, and the 2 red ones are the ones you originally got from your mom. You'll see there's 2 longer blue ones, there's a longer blue, longer red, and a shorter blue and a shorter red. When you make a new cell, if this was your DNA, each new cell has to have 1 long blue one that you originally got from your dad, 1 long red one, which you originally got from your mom, 1 short blue one you got from your dad, and 1 short red one you got from your mom. Can't be just any old 4 chromosomes. It's got to be one of each of the original 4 chromosomes has to go to each cell, because each chromosome is like a different chapter in a book. It's got different information. Each chromosome is like a different chapter in the book. It's got different information. So you've got to get one of each. There's no use getting 2 copies of the same chapter. It's redundant. So eventually we got to be able to take that cell and divide it. Notice this cell is divided properly because each new nucleus, which is the green, each new nucleus is going to have 1 long blue one, 1 short blue one, 1 long red one, 1 short red one. Each new cell is going to have the original number of 4 chromosomes. If this was a human picture, there'd be 46 in each of those green circles. But that gets too confusing. 46 is too many to track in a simple diagram. So organelles are needed to do this work. Organelles are designed to do this work of reproducing. This includes the nucleus, something called the centrioles, at least in animal cells. This is one of the structures animal cells have that plant cells don't have. Plant cells actually will involve the cell wall, which is a nice rigid structure, and they don't typically use centrioles. Animal cells, because they lack a cell wall, have centrioles. So centrioles really is just a collection of tubing, protein tubes, that provide a very rigid structure. So what they do is they help coordinate cell division. Again, I'm going to give you the bare basics right now. Second semester will get more detail. And they're only in animal cells. This is one of the distinguishing features. So you now have 4 distinguishing features. Plants have cell walls. Plants have very large central vacuoles. Plants have chloroplast. Animals have centrioles. Those are 4 key differences you'll see between plant and animal cells. There's 1 pair in each cell, 1 pair of centrioles in each cell. They come in 2s, like you see in the picture here to the right. And in this picture here, they're the little black dots. And what the black dots do is they assemble something called the spindle fibers. That's the green lines. Their spindle fibers are what literally attach to the chromosome and help pull the chromosomes apart when the cell divides. Again, we'll go into how a spindle fiber is formed later. But imagine it's like, some strings that are attached to those black dots. And those strings are going to shorten and pull those chromosomes apart. So the centriole's big role is to create the spindle fibers and then provide anchor points for those spindle fibers so they have something to pull against. In plant cells, they'll assemble the spindle fibers, but they'll use the cell wall as the anchor point. Thus, they don't need the centrioles. So there's the 2 centrioles. And what happens is eventually the centrioles cause those spindle fibers to shorten. It's almost like they're playing tug of war, and that pulls the chromosomes apart. This is how you guarantee that you get 1 of each. 1 short blue one goes one way, 1 short blue one goes the other way. 1 long red one goes to the left, 1 long red one goes to the right. 1 short red one goes to the left, 1 short red one goes to the right. And bingo, you're splitting that into 2 different cells. The centrioles are very critical for making sure you properly sort the DNA so everybody gets the proper numbers of chromosomes and gets a copy of each of the, quote, chapters. So if we go back to our animal diagram, there they are right there in almost the dead center, the centrioles involved in cell division. So that's a real quick overview of reproduction at this point. Real quick overview of reproduction. Again, we'll spend a good chunk of time second semester talking about these processes called mitosis and meiosis. But when cells reproduce, you have to divide up the DNA. We talked a little bit about that. You also have to divide up the organelles. We did not talk about that yet. That's primarily the job of the female when she produces the egg, we'll get into. And then the cell literally has to pinch off into 2. And each new cell has to have a nucleus with the exact same set of chromosomes, and each new cell has to have the proper number of organelles to function. We just did a very light skimming here. Overall, we'll get into the details second semester. So cell summary, cells have 3 main jobs. They got to make energy, which means they need food and oxygen, which is provided by the cell membrane, through the cell membrane. They carry out cell respiration and photosynthesis to create food and to break food down and produce ATP. They also need to remove waste products when they're done, like CO2 has to be removed once it's produced. We make proteins. Again, we need instructions from the DNA to do that. We use those instructions to chain together amino acids, and then using the endoplasmic reticulum and the Golgi apparatus, we're going to finish and ship those proteins to where they're needed in the body. If it was done by a ribosome that's free in the cytoplasm, that protein will fold in the cytoplasm and then be used within the cell to do whatever job it's supposed to do. And then we have to be able to make more cells, which means we need to be able to copy the DNA and divide it up appropriately between the daughter cells. All cells will do these three functions. And you can see we have specialized structures for doing each one. For making energy, we've got cell membranes, we've got vacuoles and vesicles, we've got lysosomes in some cases, and we have this thing called the mitochondria and something called the chloroplast. For making proteins, we need nucleus, DNA, RNA, ribosomes, endoplasmic reticulum, Golgi apparatus, vesicles, all involved in producing proteins. For making more cells, we've got a nucleus, we've got DNA, we've got centrioles, all involved in helping cells reproduce. So different organelles for different jobs. By having organelles, it allows the cell to divide up the work and spread the work out and have many, many different jobs going on at the same time. Sort of like what happens because of your organs. You can breathe, pump blood, digest food, and filter waste all at the same time because you have different organs to do those jobs. Same thing in the cell. It can make proteins, it can produce energy, it can get rid of waste products, all at the same time because it's got different organelles doing different jobs. Our organelles do an awful lot. That's our cellular story. This is a short segment this time, but that hopefully will give you a good refresher and review on cell structure and function. Really want you to focus on how these things fit together to do the three major jobs. Who works together to produce energy? Who and how do they work together to produce proteins? And who works together for cell reproduction? The POGILs will provide additional information that should hopefully also help fill in some gaps. Enjoy the rest of your break. Hopefully we did not take up too much of your time. We'll see you all on January 8th.